We are recording today's session and it will be available later this evening for you to watch and rewatch. We have closed captioning available, so feel free to employ that as you need to during our time together. Happy One Day 1KU. You'll notice that I and our speaker for the afternoon have our One Day 1KU background. And I don't know if you can tell, I also have my Rock Chalk Jayhawk shirt on. So we have the Jayhawk spirit with us today and always. This is a day we always look forward to. It's the university's annual 24 hours of giving. And of course, we are always intending to give to you all year long and all day long. And this is a time when so many generous folks are kind enough to give back to KU. I will have a link in the chat if that's something you want to know more about. And I will say that between 2 and 3 is our power hour. And that has a lot to do with matching generosity and matching gifts. So thank you for all of you who have given, who continue to give, and you give in so many ways, including your time with us today and participating in research and reading our newsletter. So thank you, thank you, thank you for all of those. Let's turn our attention to our topic for today. Thank you, Dr. Honey. We have your slide up on the screen. I can see it well, so I'm presuming everybody else does too. Dr. Honey is the director of the Neuroimaging Core at the KU ADRC and a research associate professor of neurology at KU Medical Center. She is the director of the Brain Imaging and Neurogenetics Lab. She earned her PhD from Oxford University in England, so she's an international scholar. And she has gotten her postdoc from KU School of Medicine. She is a member of the Society of Neuroscience, Human Brain Mapping, KU Women in Medicine, and the International Society, see, I told you she was international, to advance Alzheimer's research and treatment. So we are lucky to have Dr. Honey with us to talk about bridging imaging and biology, new frontiers in brain health research. I know that in my adventures, people very often ask exactly this question, Dr. Honey, what are we learning about imaging? Where are we going? So you are going to answer that for us today. So I will turn it over to you and thank you for being here.
Thanks so much, Kelly. That's so nice of you. Yes, I really get a lot of questions about what can you see with a brain scan? What are you doing with the brain scans? Where does it go? What are we learning? I think when people think about going to see if, when they think about a brain scan, they think about something going straight to a doctor's office and it getting read by a radiologist, and then they don't really see much of it. But I have so much exciting stuff to show you about what we are able to do with those type of scans, and really more than that, and what it's doing to advance the field when we use brain scans for research. So just a little bit about me. Everybody always likes a little background. I grew up in Texas and I always liked science. And I thought I was going to maybe go into being an MD. But when I was at Vanderbilt, I did some research in schizophrenia and I really loved it. So I went on to the NIH and did a joint program between NIH and Oxford, and really loved brain imaging and learned how to use it for research, looking for risk factors. So I came to KU Med, where my husband and I were long distance. He lived in Kansas City, so I came and married him and moved to Kansas City in 2007. And we have 3 kids. These are kind of old pictures. I've got a 7-year-old, a 12-year-old, and a 14-year-old, and so they keep me busy. And yeah, I've always been interested in looking at the brain and researching. When I was a little kid, I was talking to some high schoolers last week about what they're interested in and how that feeds it into their future. And some of the things I was showing them I loved to do when I was little were collecting bugs and doing puzzles and solving mysteries. Nancy Drew books were my favorite. I'm aging myself, but I love those. And my religion and doing Where's Waldo books. I still do those with my kids, and hunting for small changes. So that's kind of where I ended up now in my career, and looking for changes in the brain, looking for risk factors for Alzheimer's disease, I'm really passionate about it. I have multiple family members with various forms of dementia, and I really care about trying to use technology to get clear answers and to just advance our thinking. So, what do we do normally? The brain changes with aging and dementia. And typically, if you go to the doctor's office or you're talking with friends, the things that most doctor's offices will do to investigate brain changes are just memory tests, and those are important. Looking for physical changes. So sometimes we know something's gone wrong when we can't do tasks the same way. Observations about behavior. So maybe behavior is changing, and that's a way that somebody ends up knowing that something's kind of happening in the brain. Neurological exams, changes in blood work. But really, can we see something with brain imaging before any of those behavior changes come about? And we know underlying Alzheimer's, for instance, we have changes in the brain. I'm going to get into a lot of that that are caused by plaques and tangles. But all of this, a lot of this is invisible. So, really, what the field has been doing um, to really get a window into that Where's Waldo book of the brain and find these small changes is using advanced computing. And I'm not a real computer expert. I've learned a lot over the years. There are definitely more advanced people these days that really get it. And the great thing is we have an amazing team of people in our neuroimaging core at the ADRC that have backgrounds in coding and machine learning and neuroscience, and they combine all of this to do advanced computing and coding to take a brain scan from an individual and use our procedures as well as an AI read to dissect millions of data points and have scores within the day. And then we can use these images as outputs for genetic studies. And I mean, it's hard to even imagine, how does that happen? What do we do? So I'm just going to walk you through some of these visuals so you could kind of understand what we're doing at the ADRC to learn about changes in the brain. And this is kind of a rough flow, but really the brain imaging goes in, and on the left, the data goes in and turns into, a pipeline of pathway of information. The computer queries the system. We know exactly when there's a scan. It merges in with the REDCap system where we keep all of our clinical data that's still our research data. This isn't like the hospital system. And into private secure computers, we process this data for endpoints that we're interested in and make reports. And that all happens really fast and really accurately now. It used to take almost months to kind of get through that process. And now we're into the period of days and weeks, which is really amazing. And when we talk about brain structure, this is really what a scan looks like, when we send someone in to do an MRI. And we can see the structure of the brain. We can see the tissue types. We can see gray matter on the outside, kind of in the bumps and the curves. We can see white matter, which is really more white on this type of a scan. And then the CSF, which are the cerebrospinal fluid spaces, which are kind of a black color in the middle. And all of those parts of the brain can be broken up and looked at across time. So this is an example output of somebody on the left that has come to our center for 6 different scans over time. And what we are able to map out are exact changes, shrinkages, larger areas of the brain over those 6 years, down to the level of 1 millimeter cubed. And then what we do is we take that information and we are able to calculate and place them. So all of these dots you see to the right, those are people. And what we can do is we can see that when we use the right endpoints, we can find and group people really by diagnosis and other outcomes like that I'm not showing, but genetic differences or you name it. So we create various metrics based on the data that we get from the scan, and we break it into regions of the brain. We look at gray matter volume. We look at white matter hyperintensities. I'll show you a picture of what that looks like. Um, our computer programming can go in and see. You can see on the gray brain on the left, you don't really see much red. You mainly see gray and white with a blue outline. And then on the right, you can see that white matter hyperintensities are labeled in red. So you see some little red blobs inside that brain. And those are, a little bit of vascular disease. They are a marker that we're interested in, that we follow. And we can quantify the exact volume of that, in a matter of 30 minutes if we wanted to after a scan. We don't need a radiology read per se. And we can follow those over time, and we can follow very tiny changes in the brain. For instance, we are always interested in the hippocampus. It's a little tiny area right in the middle that is in charge of memory. And we have some high resolution hippocampal scans now that can actually break down, and we use, again, computer technology and a lot of expertise to break down every single subregion of the hippocampus. And we can measure those and look at differences across diagnoses, mild cognitive impairment, and Alzheimer's disease. And certain parts of that hippocampi shrink faster than others. And so we're able to follow and understand down to the level of the cell what is happening. And we can get those measurements, again, much faster than we used to be able to. Another measurement that we look at is cortical thickness, and that has to do with how many neurons you have on the edge of your cortex. And what the computer does with our help is it delineates in a person's brain, when we send it into the program, the edge of the white matter and the edge of the gray matter, and takes measurements all the way around across a whole person's brain. And we can identify very small changes in thickness that also correlate with aging and disease, and really disease type, that allow us to get to this stage of possibly, early diagnosis, and also follow positive things. So we're measuring cortical thickness before and after exercise trials and diet trials, so that we can see, well, what helps improve this. Because what we want to know is, like, can this get better? And is this something that we can improve? This is another example of ventricles and white matter. Obviously, the ventricle size changes with aging, and so that's something that we can measure pretty easily and pretty quickly. And so we can use the computer to tell us if normal appearing tissues are actually normal or not. And one of the things that we can do with another scan that's part of our regular sequence that we get on every single person is look at the axons and the white matter in the brain, and is the myelination changing. This is something that really you can't get from a radiology read. This is purely something that we do unique to the research side, where we can use computational methods to follow the water diffusion across the brain. And really what it does is it maps out all of the axons and tracks in the brain. And so we can see if more connections are made after certain tasks like exercise, which we have found and published, or with completing tasks. There's a lot of people that do games and ask me about games with aging. I really think those really help with white matter tracks and connection. There's some research there showing that those tracks increase when you do a certain activities or when you challenge your brain. We also know that in Alzheimer's disease, here on the left, that diffusion tensor imaging shows us that the tracks get smaller and they change and have less connections over time. So we can see exactly where that's happening. And that's a very advanced type of imaging scan that we do just on everybody as part of our bread and butter that we do. Um, the other type of scans we do are PET scans. PET scans, you know the difference if it's MRI or PET if you get a substance injected in you and there's a tracer. So it has a different type of procedure. So we can see that over time across the clinical spectrum, atrophy changes, which is what I just showed you with structure, but we can also see that glucose metabolism changes. Amyloid can start to accumulate, and we can track that with amyloid PET scans. And that's really been an advance in the Alzheimer's disease world, is to be able to see pathology that you used to not be able to see, until someone passed away and you did a postmortem dissection, you couldn't see this pathology, but now you can. Now you can see tau tangles and amyloid plaques and differences in glucose metabolism all in the level of the cell while the person is healthy or while the person is starting to have changes with disease, and we can measure these things, which is really amazing. And we can also place we're able to see where people fall sort of on the trajectory of how much plaque they have really quickly after a scan. So we have automated computer procedures now that are able to pass a PET scan through to get it to the level of an exact score of load of that plaque. And that's something that we can follow over time. We can give back. We give that number back in our um, when we share data back to participants if they've had that type of a scan, and we're going to be able to do even more of that going forward, which is really exciting because everybody does want to know what their brain looks like and what they have, and we're working really hard to be able to give meaningful imaging data back to everybody that comes through and is part of our studies and wants that, which, you know, outside of the ADRC, I don't know many places that do that do it to the level that we do it. And so it's just really great to be at a place that and that offers that. And it really is empowering in many ways. And we're also happy to talk with you about anything. If you ever have questions about something in a scan or something you've had done, I know I'm always happy to talk, and many of our clinicians are happy to talk about what that means. And a lot of times it's just part of aging. So I think it's important to know that not all these pictures of things that I'm showing you are scary things. Actually, a lot of times the brain is compensating for things, and so sometimes these little blobs that we show in pictures are actually good changes. And we have a lot of different biomarkers that we use to follow just healthy patterns so that we can understand prevention for disease. So what can we do with all of that data? Well, we can look at correlates of anything. We can look at how behavior relates to the brain, how cognition or disease. Like I said, we look at a lot of lifestyle and exercise studies and what that does to the brain, injury and um, and genetics really is the big piece of the thing I'm interested in. And about 10 years ago, we started wondering, like, can we combine brain imaging, genetics, and blood biomarkers to advance what we know? What if we put all the pieces of the puzzle together? Because I love puzzles. And we really, us and others have been doing this and really greatly advancing what we know. And what we do is we take blood and plasma biomarkers on the same people that get MRI and PET and their clinical information, and use a lot of imaging technology to integrate that data and ask questions with advanced statistics and really get to the level of precision medicine, because we want to know for an individual person what is the thing they need to do to stay healthy, and what is right for their brain. And we get an incredible wealth of advanced data. So those biomarkers are really all over, you know, eh, all over the place. We have hundreds and in many places thousands of biomarkers now on individuals that we use for research. And one of the things that we're trying to do is link up the brain imaging biomarkers with the blood biomarkers to understand when you get a blood test and you have a high level of A beta 4042 in your blood or NFL in your blood, does that mean that there's actually changes in your brain that we can see? So we take the same people that have blood and brain imaging and we link up the statistically link up those markers to understand exactly where in the brain those are resulting in effects. And it really helps us understand the mechanism and the pathology behind those markers so that when we explain it to individuals coming in, we really know what it's doing. And there's not a lot of places that are doing that. We've also been able to understand new genetic risk recently. We have some really interesting results that we've been working on, that I'm excited to give you a little sneak peek of. I'm not going to get into it too much because I know I want to leave some time for questions, for everybody to be able to ask. But I'll give you a brief background of like, what was the question? What are we trying to look at? We know as far as genetic risk for Alzheimer's disease that we have a group in the purple over to the left that is a causal set of 3 genes that is very, very rare that causes risk for early onset Alzheimer's. So very early, before the age of 60. And that is very rare. And then we have a lot of genes that are involved, APOE being the main one that everybody talks about. But really, I'm going to show a picture from a big study in a minute, and you'll see how many genes there are actually possibly involved. And we have these genome-wide association studies that are, what is drawing on the screen? Where they take the genes of healthy people and diseased people, and they try to look for differences in frequency of different genes in those two groups. And that's how they find genes that are called risk genes. In Alzheimer's disease, they've done an incredible number of these studies. This is data from 1.1 million individuals. And every single dot on here is a different gene hit. And you can see that above the red line, there are hundreds of genes involved. And not all of those have a function. Some of those play different roles. Some of those things may have to do with aging. They may have to do with other risk factors for other diseases. But the one thing that really stands out most of the time is chromosome 19. There's always a lot of hits on that one because APOE is on there and it's a known risk factor. But one of the things we've done in collaboration with some researchers at Duke is we've looked at what are the neighboring genes to APOE. There's 2 genes on either side that also show up on all of these studies, but no one looks at them. And what if those were the genes that actually are making a really big difference? And one of these genes, TOM40, I've been very interested in for a while because TOM40 is on chromosome 19. It's next to APOE, and it also governs a very important complex in the mitochondrial membrane, and it allows the influx of amyloid beta into the organelle. And that's really interesting because amyloid beta or plaques are a big part of the Alzheimer's disease process. So it gives us a clue that there is probably something having to do with TOM40 in Alzheimer's disease. There's also a SNP, or a really small change in TOM40, that's associated with AD risk, but not many people have looked at it. So I had the thought, a couple of years ago with some of our colleagues of why don't we look at our really rich, amazing data set with all of these neat neuroimaging markers. We can look at this SNP in individuals that have no Alzheimer's and see if it affects the brain at all. So we took a big sample of our individuals and did our fancy imaging and with our fancy colored scans and looked at thousands and thousands of data points across the brain. And what we found was that there is a relationship between that particular gene and some of the changes that we see that happen really, really early on with Alzheimer's. And we saw this in people that don't even have Alzheimer's. So it gives us a window into the function of the gene. And then we also found that there are relationships between that gene and differences in males and females and the limbic area of the brain or the memory area of the brain. And that females seem to have more atrophy with aging. And this has really gotten me on the subject of really wanting to understand how genes affect men and women differently and what we can do to maybe possibly intervene differently with men and women for prevention. So that's going to be some of my future research. And then another study that we did that's recently published that's very exciting has been to look at all three of these genes together. And what happens if we look at them all together? One of my colleagues looked at tens of thousands of individuals with various combinations of these genes. And what he found that was that this one little red line in the middle, that's if you have both E4 alleles, you have pretty high risk. That's what everybody always says. It turns out it's not that high of a risk compared to if you have the other genes too, and you have their risk alleles. It turns out that triples your risk. And no one really knows this. And so no doctor's offices test for these genes. They don't really look at them. And we also know that those other genes can be protective, those neighboring genes. And so what we're trying to understand and what we recently published is looking at those genes across all of our brain imaging markers to understand how they're interacting for risk there. So that's a little sneak peek. I'm really excited about what it means for our understanding of risk for Alzheimer's disease. And I really think that brain imaging has been key for our understanding and that we're growing leaps and bounds with our technology. And then we hope to get even more close to personalized medicine and prevention for everybody so that we can prevent this disease. And thank you to all my collaborators and everybody that did all this work, which is a lot of people. This is not me. Thanks, Kelly.
Thank you. That was great. And I don't know how much you saw, Dr. Honey, that there are some really excellent questions in the chat.
Sure.
So if I can point you there, and if you're willing to, I think we might need a little more than 2 minutes. So if you're willing to stay on and answer
Yes,
some
I'm
of these
happy
questions.
to.
Okay, well, let's go to the top, just sort of in the order that we've gotten them. And we have, how does one qualify for these scans? And from the same person, I've been in a healthy brain cohort for 8 years. Will this, I think, imaging be
Yeah.
included in my test?
Well, I guess I would ask if you had scans, and if you were in our cohort at the ADRC, and if you are, then we have this, we have all this for you. And at some point, you probably have been given some feedback of some of this data, I would imagine, if you're part of our cohort. I know that at some of the next upcoming visits in the next year, they'll be able to share even more as we've really advanced our markers that we're getting out and giving back. So I think, if you haven't had any scans as part of our cohort, um, they will find a way if you want it. I think, when you go in, you can, not everybody wants to get a scan, but if you want scans, I would just encourage you to ask. I ask your physician, whichever one you're with, because we are even today, as part of One Day One you collecting more funding to offer this neuroimaging technology, so that it can become more available for people that aren't just in a study, but that want to get a brain scan for these purposes. So hopefully that will be possible. Let's
Great
see.
news. Okay, let's see. Next, we've got when do you think tau PET scans will be available in a clinical setting versus research setting?
Yeah. That's a good question. I know that we're going to be offering tau PET scans starting this year in the next month or two as part of our Clarity project, which it, you can sign up to be a part of that. That will offer amyloid and tau PET, and then our research scans will start collecting tau PET as part of our standard protocol, and I think it's 2026. As far as the clinical scans, I do believe that you can request them, but that's a good question. I'm not sure how they're funded. They are still expensive, but I know that Jeff Burns is getting them on some people. I don't know if it's always as part of a study. That may be how people are getting them paid for, but I know that it's possible.
Okay. All right. The next question we have is there a one-stop method to find out if a person is a candidate for any of the KU dementia studies, if they're not having problems, but do have a family history?
Sure. So we have a lot of people, and family history is my favorite area of research. And we have been trying for a while to come up with a family history cohort study that would be younger ages, because we have a lot of midlife, 40 to 60, that want to come. 60 and above, you can come and be a part of our healthy aging cohort um, with your family history and any interest and be a part of that. It does require that you be a part of the study. So there are yearly visits and brain scans, especially on your first visit. You get we do brain scans on all first visits, and then every other year you would get scanned, and so as well as get data back, and learn about other studies as you're a part of that. So that cohort study is really about monitoring um, and kind of collecting a baseline and getting data. So I would definitely look into that, because
Okay.
that's exactly the right reason that we want to have somebody come in.
Yeah, and I think that that may have been actually a partial answer to our next question, which is that my mother has been a patient with the ADRC since 2017 and has never been offered imaging. Is this only available as part of a study?
Yeah. So um, yes, if unless she's enrolled in the cohort, which honestly, there just hasn't been a lot of funding for research scans outside of us funding Madi paying for that scan, and that's probably why it wasn't offered. Although I would say to always ask the physician, because they can, depending on whatever kind of insurance you have, they can order scans now as part of the diagnostic procedure or for any concerns. And so scans are used now for that. They weren't really even 5 years ago. So I think things have changed, and there's options either through the physician visit or through our clinical cohort or a particular study that all would include imaging.
All right, our next two questions, I'm going to put them together because I think they're part of the same answer, I hope, based on my perspective, Dr. Honey. And so the question is, my mom is having an amyloid PET scan next week. She's telling me they think she had a mini stroke, and this scan is to look for that as well. Are amyloid PET scans only for dementia detection, or can they see stroke damage as well? And then the next person is asking, what effect does a stroke have on the brain? So if you could maybe
Yeah,
answer
that's a great
both of
question.
those. Yeah.
And actually, when people come in, they do sometimes do a couple different type of scans to rule out a stroke, if they're because some of the symptoms that happen with aging and various types of dementias, they sometimes look like a stroke. And so what they do is they'll just do several things. So they can see, was there a stroke or not? And that would be on the CT scan that happens when you get the PET. They do a CT scan, and that would be where they can usually see those type of bleeds. And then the PET on top of that would show, is there So if you think about it, plaques or amyloid plaques are kind of like scabs that are going to damage. They are not part of a stroke, but if you have had one, or if you have even sometimes had, traumatic brain injury, there can be plaques and tangles that are part of that brain trying to heal itself. So they would use the information for that as well. And then they probably would do um, some types of resolution scans to see if there's any areas that have been damaged, like in a mini stroke. And so it's not, I don't always hear about that, but I do think getting all the types of scans is probably a really good thing, because then you get a real answer for what you actually have. So they're not treating you for Alzheimer's when you actually had a stroke, or a stroke when you really have Alzheimer's, because those are very different pathways. I hope that
Right.
helps.
Okay. Next question is from, I hope you don't mind, one of my colleagues. I mean, I know you don't mind. I hope my colleague doesn't mind me saying she's one of my colleagues. And she's curious about whether there are many or any genes that can increase or decrease risk for Alzheimer's, depending on other factors. So, for example, a gene that increases risk in one population specifically, but might decrease risk in a different population. Does this ever happen?
Yeah, it does happen quite a bit. And we have this bulk of data in our country that is primarily Caucasian and European ancestry, that has given us a great amount of information about that particular group with risk and APOE. And what our science field has been trying to do in the last 5 years going forward is to collect on other demographic groups and ethnic groups, and so that we can really understand why things are different there, because the APOE gene is different with African Americans, for instance. And so it appears to have a lot less of a risk factor. And um, and we don't know what other genes are different like that too. And we really need to understand that, because you don't want to say it's risk for one and not for somebody else, right? And so we're really there's thankfully a large amount of growing databases to be able to look across all different groups and understand risk across their groups differently, and things like neighboring genes and how those are interacting. And yes, there are protective genes too. So we don't talk about that as often, but the APOE2 allele is very protective. And then there are also protective mitochondrial genes and aging genes that people have that are keeping them from getting the disease. And so that's great too.
Wow, that's so encouraging to hear. Thank you.
Yeah.
Okay, gosh, these questions are coming in. Okay. This one I think refers to your slides, Dr. Honey. And the question is asking about the table that shows the risk with and out MA, which I don't remember what MA stands for. I'm sorry. And it was near the end of the presentation. Um, and
Yeah. I
yeah.
don't know what MA stands
Oh,
for.
good. It's not just me. Okay. The person
Okay.
who asked that question, if you're still here and you want to tell us what MA stands for, that would help.
No,
Uh,
I didn't know either.
oh,
Sorry for the
good.
that's why I'm asking, so I could find
Oh,
that
good.
out.
Thank
With
you.
and
Okay.
without.
It was like the
And
second or third to last slide. If you don't know offhand, that's fine. I was just trying to follow along at home.
oh, you're right. Yeah. Um, yes, I should have clarified. That MA has to do with their definition of a compound genotype, which I didn't list the definitions. And so I'll have to send you the paper, or um, have Kelly share it out. And
Yeah.
I'm always happy to do that. I list the citation. It's Kolminsky et al. 2022, and I'll put that in the chat. Um, or I'll try to find a link to share so you can learn a little bit more about that, because it's a really good study.
Okay, very good. And I'm just putting in the chat, if you want what Dr. Honey just offered, send an email to my alliance at kumc.edu and we'll share that. Okay, let's see. We've got, let's see, I've got a couple things that were just to me. We've got some thanks, Dr. Honey, from people who are saying that they better understand the cohort results over the last year. So that's great to hear. And we've got a question about whether cancer is still a disqualifier for participation in the studies.
I don't know. I don't think it would disqualify from all studies. There's a lot of different people with different histories in some of the studies. So I think sometimes they're a little more strict with pharma studies. Most of the research studies I'm talking about are not in that line. They're more lifestyle studies. So I would definitely reach out. Kelly, do you have a contact for who people can ask about the various studies and then also how to join the cohort?
I do. I'll put that in the chat.
Okay.
If you will take, then I'll put some information about, who to contact. And then I think our last question, there's a bunch of links in the chat, so make sure you see those. And then our last question is about a 26-year-old son who had some paranoia and psychosis, but not memory loss. And can MRI scans be helpful in diagnosing and treating that?
Yeah, so my original work when I was doing my PhD was at the NIH doing schizophrenia and psychosis research. And really, I learned a lot of these techniques on that subject group because they are using very similar things. Now, I don't know if there's a such thing as, we have these 21 nationally funded Alzheimer's centers, and we're able to leverage as a team all of this data and put it together. I don't know if they have the same thing for mental health. I really hope that they do, but I, and I hope that they've gotten it to the level of practice. Unfortunately, it's just what I found with some neurologists and some psychiatrists, and I don't want to belittle anyone because I don't have that degree and I'm not doing that job, but they don't have these types of scans as part of their protocol. And so I do feel like some things are missed. And so, I hope that you can find somebody that looks into it, because I know even 17 years ago when I was doing psychosis research that we had a lot of these techniques already. So somebody has it somewhere. Okay, I see another one about family history of Alzheimer's and sleep apnea and numerous sleep tests, blood tests and computer testing many years ago. Oh, it sounds great. You're improving your diet. Great job on not having alcohol for 2 years. Wow, that's amazing. And athletic. So good for you. I guess there wasn't a question, but that's really what we should all be doing. Bravo.
Yeah.
That's a great, I guess that was sent to me. So thank you. Sorry for reading that out loud. I still getting used to some of the Zoom. But I think having an interest in your own health and being able to push yourself to, Jeff Burns still says to get out and do 10,000 steps a day. I have a lot of trouble with that. I think if you can even try 6,000, 5 or 6,000, let's start there and try to get out and get some fresh air and get rid of those foods that are, just are not working for your body. Those are really right now, I always am telling people better than the drugs. So I think, you have the power to change some things more than pharma does. I mean, you're talking to the researcher though. So, I don't want to discourage anybody. There's some great products out there, but I just think there's a lot that we can still do. So, hopefully that will resonate.
Oh, definitely. I think Dr. Honey, your stats, your creds as a researcher go a long way here. So thank you for sharing with us. Thank you for staying a bit past our time to answer all these great questions. Thank you to all of you for being here this afternoon. Remember that it's One Day 1KU until noon Central Time tomorrow. I put the link in the chat, and so you can check it out if you would like. And know that your support of One Day 1KU helps us to do exactly this and to help fund a lot of the research that Dr. Honey mentioned, especially she mentioned that we could have more imaging if we had more money. She did not tell me to say that, but it's nice to know. So thank you again. We appreciate your time,
Thank
Dr. Honey, and we appreciate all of you being here. Next week is the end of February. I'm probably not alone in saying February can take itself out of here with all this cold weather. So I will be happy to see the end of the month. And our speaker next week is a couple of representatives from a program that will talk about support for Missouri caregivers. Now, all you Kansas people, do not think that you shouldn't come. You should still come, because they are going to share with us information about how to support caregivers in lots of situations. They just happen to be over on this side of the state line. So there will be a little bit more of a focus on Missouri, but you will still find it valuable to come and hear what they have to say. So that's next Thursday, February 27th at 2 o'clock. You're welcome to the person who said, thank you for Missouri, my pleasure. We know My Alliance touches people, 8,300 people now all over the world. So we are always looking for resources available to all those different groups. You'll see more information about that in Monday's issue of My Alliance. And I hope you all have a great rest of the day. I hear it's going to be in the 60s in Kansas City next week. Yes, I'm happy about that. And I think we can wrap up. Yes, thank you so much. And we'll see you next time. Bye, everyone. Thank you, Dr. Honey.